title of my talk is The Strategies Towards Improved Prediction of Drug Response in Acute Myeloid Leukemia. And I am currently Associate Professor of Biomedical Sciences and the head of the Cancer Biology and Therapy Lab at the School of Applied Sciences at London South Bank University in the UK. So I'm going to talk to you today about two strategies that we have been using to better predict drug responses in AML. One of them is by generating or creating a bone marrow mimicking microenvironment culture conditions for drug testing. And the other one, which I'm gonna touch upon lightly, is the proteomic profiling. So just a little bit background about pediatric acute myeloid leukemia. Actually, I worked on both pediatric leukemia as well as adult leukemia. So the first strategy, we used pediatric acute myeloid leukemia cells. And just as a background, the AML incidence of all diagnosed pediatric leukemia is about 20%. And the 5-year survival rate for children below 15 years of age at diagnosis is about 66%, of course, varying based on the AML subtype. And the current therapy of AML is chemotherapy, which is the first-line therapy, bone marrow transplantation, and targeted therapy. The cost of therapy per patient in America ranges from $250,000 to $1 million, with an average of about $500,000. And the overall, actually, survival rate for patients with relapsed refractory AML is below 30%. And this is the fraction of the patients that we would like to target and focus on in order to slightly improve, if we can, this survival rate. So the challenges faced by hematologists, among the key challenges actually in this era of new drugs, is the timely identification of actionable mutations at diagnosis and relapse, as well as deciding which drug to use among several therapeutic options. So to address these two challenges, we have followed this workflow. So we, at this work I'm showing now is from the time I was working in America, and we were in very close contact with the children hospital in Phoenix. So this is the workflow that we were using, and it is similar to a workflow that was been used by the lab of Olli Kalaniemi in Finland, which is we get the bone marrow samples at diagnosis and relapse, and then it has two fates. One of them is the drug testing, what we call the drug sensitivity and resistant testing. And what we have tried to optimize in this technology is to perform the experiments in 3D and in hypoxia. And I'll show you why in the next slide. And the second pathway, the things that we do, actually the experiments that we do with the samples, is to generally do a whole exome sequencing and RNA sequencing in order to obtain information about the changes in gene expression and the mutations. But what our lab tried to do is to add a third dimension, which is proteomic profiling, in order to try to obtain additional information so that we can have a comprehensive picture of what's happening in each individual patient. And by combining the results of these two strategies, we can get more understanding of the biology of the disease, as well as the drug sensitivity and resistance, which we present the data to a tumor board for the rapid introduction of therapies. So the first strategy was to develop an in vitro model that mimics the bone marrow microenvironment to better predict patients' response to therapy. And what is in most labs, scientists or researchers grow cells in 2D conditions. And then this is not really the 2D, and the atmospheric oxygen is 21%. And the 2D is just a plastic that, therefore, the cells are not very reflective or representative of what's happening in the physiological bone marrow. This diagram here shows you what's happening in the bone marrow. So there is low oxygen, hypoxia. There are endothelial cells, leukemia stem cells, RBCs, fat cells, macrophages, so many other cells, of course, stromal cells as well. So what we try to do is to create this, what we call bone marrow mimicking conditions, in which we grow the same cells in 3D structure, as well as try also to co-culture some stromal cells, and we could actually distinguish between the leukemia cells and the stromal cells using live cell dyes, like green for stromal cells, red for leukemia cells. I'm going to show you today some results from the hypoxia study, as well as the 3D studies. So our workflow for drug sensitivity and resistance testing is as follows. So we have those 384 well plates, the compound library, and we have anything up from starting from 50 different drugs in 5 different dilutions. And then we print the compound library into other plates using the ATS acoustic dispenser, which is a robotic machine. And then after that, we can freeze or store the printed cells in -80 degrees for as long as we will. But when we want to do the experiment, we then thaw the plates with the compound library and then add the cells. We can add them actually manually, or we can use the multi-flow dispenser, which has a semi-robotic instrument. And then we grow the cells either in normoxia or hypoxia in 3 hours, for 3 days, followed by drug cell viability using cell titer glow, as well as further mechanistic studies as well when we get meaningful results from the viability studies. So this is just to show you, and all these results are confidential, please, not for sharing. This shows you the hierarchical cluster of the different drugs that we used against more than about 12 different cell lines. The white columns here show you the cells grown in normoxia, and the black columns shows you the cells grown in hypoxia against about 50 different drugs or compounds. Some of them are FDA approved, some are investigational drugs. Excuse me. And we have in the red, like here is the DMSO. We always have to have a negative control with the dimethyl sulfoxide. We treat the cells with dimethyl sulfoxide. So when the cells do not react to anything, they are as red as the last row here in this diagram. The blue color shows death. The more the blue means more death, and the more towards the 4+ shows viability. And any colors in between show, a range between life and death, between the red and the blue colors. So we have, out of the 50 different compounds we used, there were about 4 compounds that were that showed a dramatic difference between viability in hypoxia and normoxia. So we started to focus on one of them. I'm going to call it X, drug X or compound X. And we have actually used this drug response assay in order to confirm our the experiments I showed in the previous slides. And indeed, using the compound X, we had, this is the percentage of viability, and this is the log nanomolar of the compound. And the green is normoxia and the red is hypoxia. And you can show that the cell, you can see that the cells die much later than in normoxia. So it's more than 3 or 4 times fold the difference in effect. So this here, we wanted to see why the cells don't die in hypoxia actually. So we did apoptosis and apoptosis assay using flow cytometry. It's annexine 5 and cytox red. And these here, the upper panel shows the cell in normoxia and the lower in hypoxia. These scatter plots shows cells in DMSO, whether normoxia or hypoxia, the negative control. And these cells here show the treatment of the compound X using either 1 micromolar or 5 micromolar in normoxia, and either 1 micromolar or 25 micromolar in hypoxia. So it shows you that using 1 micromolar of the compound in normoxia kills about 95 combined together. This is the early apoptosis and late apoptosis combined together. About 95% of the cells die after treating the cells only with 1 micromolar in normoxia. while just 17% or 27% die using the 1 micromolar of the same drug in hypoxia, which is more or less the same effect of the DMSO. Cells have not died except even when we use even 25 micromolar of this drug in hypoxia, it still kills about 86 or 90 something, while here it kills more than 100% of the cells. So we wanted to understand why is this happening. So we performed RNA sequencing of the cells grown. This is a pediatric leukemia cell line with a FLT3 mutation, which is a very common mutation for acute myeloid leukemia. So here, this shows this is the result of the RNA sequencing performed after treating the cells with this drug, drug X, in normoxia or hypoxia, and we collected the cells after 9 hours, and this timeline has been optimized in order to screen for the differentially expressed genes. And as you can see, without even seeing the labels, this column or this row here shows quite different pattern than the other 3 rows. And indeed, this is this row is the one, the cells that has been treated with drug X in hypoxia, while these ones have been treated with the vehicle, the DMSO in normoxia, and these are the vehicle in hypoxia. And this one in the middle, the row in the middle here, or the second one from top, shows actually the differentially expressed genes of the cells that after treatment with drug X in normoxia. So there is a stark difference between the cells when they're treated with the drug in normoxia and in hypoxia. We also did some pathway analysis, and we found that these are the pathways overrepresented in hypoxia versus normoxia regarding the vehicle. This is the pathways overrepresented by drug X in normoxia, including cell cycle, p53 damage pathway, and so on. And these are the pathways overrepresented by the X drug in hypoxia, which includes the JAK-STAT signaling pathway, as well as the stem cell markers or the stem cell activated pathways. So it's different pathways that are activated when I treat the cells with this compound in hypoxia versus normoxia. So we also performed the same experiments in 3D, and this is just one slide to show you that in 3D, when we have grown the cells, it's a Western blot to show you that when we have grown the cells in 2D versus 3D, in normoxia versus hypoxia, untreated versus treated, we found that in 3D and hypoxia, treatment of the cells, the MV411 cells, with this compound causes stabilization of the Mcl-1 protein, which is an inhibitor of apoptosis, compared to the same experiment when it is carried out in normoxia. Normoxia, the Mcl-1 completely disappears, and also compared to 2D. So from this, we conclude that, by the way, we also optimized we did the same experiment using patients' mononuclear cells derived from the bone marrow. And it have taken us a while. Maybe this also links to what Dr. Jihan just presented about optimization of the stem cells and so on. So it took us a while. We performed the exact same experiment using patient-derived bone marrow cells, and we could culture the cells in culture with a viability of about from 75 to 88%, when we got similar results as well. But I decided not to show this slide because it's from patients and we haven't published that yet. So the summary from this part of the talk is that hypoxia and 3D structure do alter drug response in AML cells, and the proposed 3D assay can be also used for high throughput drug screening, as well as that the resistance to drug X is associated with stabilization of Mcl-1, and the bone marrow mimicking culture conditions in drug testing may lead therefore to more accurate predictions of therapeutic outcomes. So the second strategy, I'll show just 2 or 3 slides of that, was that we, as I mentioned in my workflow slide, we tried to add another dimension in addition to the genomic and transcriptomic profiling, which is the proteomic profiling. And the rationale for using proteomic profiling is that the genomic-based stratification of patients with AML is not sufficient to predict their response to therapy. So the goal of this pilot study was to determine the proteomic signatures in adult patients with AML associated with the traditionally classified clinical subtypes. And this figure here shows you that this figure is from a publication from the lab of Suhail Mishinchi in Seattle, and it shows the molecular landscape of pediatric acute myeloid leukemia, which reveals that, for example, some mutations such as KRAS is very frequent in infants, and it goes down from infants through a childhood and adolescence until adult. The other way around for the NPM1 mutation, it is more prevalent in adult AML versus infant AML. So what we have done is that in collaboration with the lab of Brian Drucker in Oregon Health University, we have obtained about 32 different adult acute myeloid patient samples. And we had different, they represented different acute myeloid leukemia subtypes. For example, those with the mutated NPM1, CEBPA mutations, myelomonocytic, therapy-related, and the so-called not otherwise specified leukemia. We were interested in the therapy-related acute myeloid leukemia because it is not characterized, and it's not well characterized, and it is rare, and it is associated with poor prognosis. So we have done LC-MS-MS proteomics using the Orbitrap Fusion Lumos, and we could found the significantly enriched pathways by subtype. So these are the different subtypes: the mutated NPM1, CEBPA, myelomonocytic, therapy-related, and NOS, or the not otherwise specific leukemia. We found two specific pathways that are enriched in common between the therapy-related AML and the NOS AML, and these were the cancer drug resistance pathway and the chondroitin sulfate degradation pathway. In addition, we could also, although most of the, as I mentioned, we had about 4 different adult leukemia subtypes, when we performed the proteomic profiling in order to find or identify kinase signatures, we found that most of the patient samples clustered together, except actually 2 patients that belonged to one subtype called the NPM1 mutated subtype. 2 of these patients, as well as 1 patient that was belonged to the CEBPA subtype, they both actually showed very much kinase expression levels and hierarchical clusters closer to subtypes with poor prognosis, because actually the NPM1 and CEBPA are subtypes with good, favorable prognosis. But only 3 patients out of these 2 subtypes actually clustered more with the poor prognosis subtypes. So we can, and especially specifically for the expression of some kinases, including the PKN1 and the FGR. So then we had also shown the same patients to cluster also differently and together with the therapy AML and myelomonocytic AML in another experiment, which was the unsupervised clustering of significantly abundant proteins, highlighting molecular mechanisms more akin to these subtypes. So to conclude, our proteomic profiling, this part of the talk, our proteomic profiling actually may contribute to reclassification of patients into other risk groups or AML subtypes, as well as better select targeted therapy based on activated protein pathways. With this, I mean with the kinase signature that we identified, as well as predict or better predict response to therapy. So the conclusion of these 2 parts or these 2 strategies is that clinical application of both strategies presented here may lead to more accurate predictions of therapeutic outcomes, whether in pediatric or in adult AML. With this, I thank you very much, and I would like to thank the lab from which I got the samples for the proteomic study, the Oregon Health and Science University Brian Drucker lab, as well as my colleagues at Phoenix Children's Hospital and TGen. you.